Hello everyone, welcome to our first session in this course. Today we're starting with Module 1, General Introduction to Medical Biochemistry. I'm excited to have you all here. In this presentation, we'll explore the fundamentals of biochemistry as it relates to medicine. We'll look into cell structure and functions in Study Session 2, focusing on the biochemistry of macromolecules. So stay tuned as we uncover how these essential molecules support life and health. Let's get started. A cell is the smallest unit that is capable of performing all the necessary life functions. Think of it as the basic building block of all living organisms. So when we ask, what's a cell? We're really asking about the fundamental piece that makes up all plants, animals, and even microorganisms. Even though cells are tiny and often invisible to the naked eye, they are incredibly complex and efficient. They carry out essential tasks like obtaining nutrients, producing energy, and eliminating waste. Understanding cells helps us see how living things grow, develop, and stay alive. This is a crucial concept because everything in biology revolves around these tiny but mighty units. Let's take a closer look at the cell timeline. This slide highlights the key stages in the life cycle of a cell, which is fundamental to understanding how biological processes develop over time. First, we see the process starting with cell growth, where the cell enlarges and prepares for division. This is a crucial phase because the cell needs to reach a certain size and have all the necessary components before dividing. Next, we move into the DNA replication phase, where the genetic material is duplicated. This step is vital for ensuring that each new cell receives a complete set of genetic information. Following replication, the cell enters mitosis, the process of actual division. During this stage, the cell's nucleus divides, and then the cytoplasm splits, resulting in two identical daughter cells. Finally, after division, the cycle begins anew with each daughter cell growing and preparing for their own division cycle. Connecting this to our overall theme, understanding this timeline helps us appreciate how cells maintain continuity and function over generations, which is essential for growth, development, and healing in living organisms. Schwann and Schleiden were two pioneering scientists whose work laid the foundation for what we now call the cell theory. They proposed that all living things are made up of cells, which was a groundbreaking idea at the time. The key point here is that cells are the smallest units of life that can still perform all necessary functions. This means that everything alive, from tiny bacteria to complex humans, is organized around these basic building blocks. Another important concept is that all cells come from pre-existing cells through a process called cell division. This idea challenged previous beliefs and helped us understand how life propagates and maintains itself. So when we talk about cell theory, we're essentially highlighting these core principles: that cells are the fundamental units of life, and that they arise from other cells. This theory is central to understanding biology because it connects all living organisms through their cellular makeup. It also ties into our overall theme of understanding life at its most basic level. Let's take a closer look at eukaryotes and prokaryotes. These are the two main types of cells that make up all living organisms. Eukaryotes are generally more complex. They have a defined nucleus that contains their genetic material, and they also have various membrane-bound organelles that perform specific functions. Think of eukaryotic cells as organized and compartmentalized, which allows them to perform more specialized tasks. On the other hand, prokaryotes are simpler in structure. They lack a true nucleus, meaning their DNA floats freely in the cell's interior, and they don't have the same kind of membrane-bound organelles. Bacteria are a classic example of prokaryotes. Despite their simplicity, prokaryotes are incredibly diverse and resilient, thriving in environments where few other organisms can survive. Understanding these differences is key because it helps us grasp how life has evolved and diversified. The complexity of eukaryotic cells enables multicellular organisms, including humans, to develop specialized tissues and organs. Meanwhile, prokaryotes, simplicity and adaptability have allowed them to colonize almost every environment on Earth. So, in the broader context of biology, recognizing these cell types helps us understand the fundamentals of life, from microscopic bacteria to complex plants and animals. Now, let's take a closer look at some different types of cells. Here we have examples like amoeba, muscle cells, nerve cells, sperm, cheek cells, red blood cells, and white blood cells. The human body is astonishing because it contains about 100 trillion cells. And what's even more fascinating is that there are over 200 different types of these cells, each one specialized to perform specific functions. For instance, muscle cells help us move, nerve cells transmit signals, and red blood cells carry oxygen throughout our bodies. These cells don't stay the same. They multiply to produce copies of themselves, allowing our bodies to grow, heal, and replace old or damaged cells. Understanding these different cell types and their functions is key to appreciating how complex and efficient the human body truly is. Each type plays a vital role, working together to keep us healthy and functioning smoothly. Let's take a closer look at an idealized animal cell. This diagram helps us visualize the key components that make up the cell and how they work together. As you can see, the cell membrane surrounds the cell, acting like a gatekeeper to control what enters and exits. Inside, we have the nucleus, which is like the cell's control center, containing genetic material and directing activities. The cytoplasm fills the space around the nucleus and holds other organelles in place. Moving on, the mitochondria are often called the powerhouses of the cell because they generate energy. The endoplasmic reticulum, both rough and smooth, plays a role in protein and lipid synthesis. The Golgi apparatus is responsible for packaging and distributing proteins. And there are other important structures like lysosomes, which help break down waste, and the cytoskeleton, which provides support and shape. All these components highlight how complex and organized an animal cell is. Understanding these parts is fundamental because they work together to keep the cell functioning properly, which ultimately supports the health and activity of the entire organism. This slide ties into our overall theme of understanding cellular structure and function, which is the foundation for studying biology at a deeper level. So, when we look at a cell, we see that it can be divided into three main parts, and understanding these parts is fundamental to grasping how cells function. Today, we'll focus on the overall cellular anatomy, which provides the foundation for everything that happens inside the cell. First, there's the cell membrane, which acts like a gatekeeper. It controls what enters and exits the cell, maintaining a balanced environment inside. Think of it as the cell's security system. Next, inside the cell, we have the cytoplasm. This is the gel-like substance that fills the cell and holds all the organelles in place. It's where many of the cell's activities occur, like manufacturing and energy production. Finally, the nucleus is often called the control center. It contains the cell's genetic material, DNA, and coordinates activities like growth, metabolism, and reproduction. Connecting this to the overall theme, understanding these three main parts helps us see how cells operate as tiny, highly organized units. Each part has a specific role, and together, they keep the cell functioning efficiently and responding to its environment. The plasma membrane is essentially a phospholipid bilayer that surrounds the cell, and this structure plays a crucial role in protecting the cell's interior and its organelles. Think of it as a flexible yet sturdy barrier that keeps everything inside safe from the external environment. One of its key functions is to separate the extracellular fluid, which is the fluid outside the cell, from the intracellular fluid inside the cell. This separation is vital because it allows the cell to maintain a stable internal environment, or homeostasis. The plasma membrane also acts as a gatekeeper. It regulates what substances can enter or leave the cell, ensuring that nutrients get in, waste gets out, and harmful substances are kept at bay. This selective permeability is essential for the cell's survival and proper functioning. Additionally, the membrane facilitates interactions with its surroundings and neighboring cells. This communication is vital for processes like tissue formation, immune responses, and cell signaling. So overall, the plasma membrane isn't just a barrier. It's a dynamic structure that supports the cell's protection, communication, and regulation, tying into the bigger picture of how cells function as the building blocks of life. Now, let's take a closer look at the cytoplasm. This is the jelly-like substance that fills the inside of the cell and is enclosed by the cell membrane. Think of it as the cell's internal environment, providing a supportive and somewhat flexible medium. Its main role is to serve as a platform where all the chemical reactions necessary for the cell's survival and function can occur efficiently. Without the cytoplasm, these reactions wouldn't be able to happen smoothly, which makes it essential for maintaining the cell's health and activity. Understanding the cytoplasm helps us appreciate how the various parts of the cell work together seamlessly to keep the cell alive and functioning properly. The cell nucleus is the most noticeable organelle in a eukaryotic cell. It's essentially the control center of the cell, housing the cell's chromosomes, which contain our genetic information. Inside the nucleus, almost all DNA replication and RNA synthesis, or transcription, takes place. This is where the cell's instructions are copied and used to make proteins. The nucleus itself is spherical and is separated from the cytoplasm by a double membrane called the nuclear envelope. Interestingly, the nucleus is also involved in ribosome synthesis, which is vital for protein production. Now, here's a fascinating fact. 2 meters of human DNA can fit into a nucleus that's only about 0.000005 meters across. Think of the nucleus as the cell's command hub, tightly packed yet highly organized, overseeing all the critical activities that keep the cell functioning properly. The plasma membrane is essentially a phospholipid bilayer, which serves as the fundamental structure of the cell's outer boundary. This bilayer is made up of several key components that work together to maintain the cell's integrity and facilitate communication with its environment. First, we have lipids, which include phospholipids, glycolipids, and cholesterol. Phospholipids are the main building blocks, forming the bilayer with their hydrophobic tails and hydrophilic heads. Glycolipids are lipids with carbohydrate groups attached, playing a role in cell recognition and signaling. Cholesterol, on the other hand, helps modulate the fluidity and stability of the membrane, making it flexible yet sturdy. Next, proteins are embedded within this lipid bilayer. These are classified as either integral or peripheral proteins. Integral proteins span across the membrane, often functioning as channels or transporters, while peripheral proteins are attached to the membrane surface, assisting in signaling and structural support. Finally, carbohydrates are also part of the membrane structure, primarily in the form of glycoproteins and glycolipids. These carbohydrate chains are crucial for cell recognition, communication, and adhesion. Putting it all together, these components collectively form a dynamic and versatile membrane that not only protects the cell, but also actively participates in its function and interaction with the surroundings. This complex structure is fundamental to understanding how cells communicate and respond to their environment. Let's take a closer look at some of the key membrane lipids, starting with phosphatidylcholine. This molecule is one of the most abundant phospholipids in cell membranes and plays a crucial role in maintaining membrane structure and fluidity. Next, we have phosphatidylphenolamine, which also contributes significantly to membrane integrity and is involved in membrane curvature and fusion processes. Then there's phosphatidylserine, a lipid that's important not only for membrane structure, but also for signaling functions, especially in apoptosis. Moving on to phosphatidylinositol, which is essential in cell signaling pathways, particularly in processes like cell growth and differentiation. We also can't forget sphingomyelin, a sphingolipid that contributes to membrane rigidity and is involved in signaling as well. All these lipids are categorized under membrane lipids, specifically phospholipids, which are fundamental to forming the bilayer structure of cell membranes. Understanding these lipids helps us grasp how membranes are dynamic and versatile, supporting functions like communication, transport, and protection. This overview ties into our broader theme of exploring membrane composition and function, emphasizing how each lipid plays a unique and vital role in cellular life. Today, I want to talk about the behavior of phospholipids in aqueous solutions. As you know, phospholipids are fundamental components of cell membranes, and their behavior in water is key to understanding membrane structure and function. When phospholipids are placed in water, they tend to spontaneously organize themselves. This is because of their unique structure, a hydrophilic or water-loving head and hydrophobic or water-fearing tails. The heads are attracted to the water, while the tails try to avoid it. This leads to the formation of structures like micelles or bilayers. In micelles, the tails are tucked inward, away from water, forming a spherical shape. In bilayers, the tails face each other, creating a double layer, which is the fundamental structure of cell membranes. This self-assembly is driven by the need to minimize unfavorable interactions between the hydrophobic tails and water. Understanding this behavior is crucial because it explains how cell membranes form and maintain their integrity in aqueous environments. It also highlights the importance of phospholipid properties in membrane fluidity and functionality. So, in essence, the behavior of phospholipids in water underpins much of what we know about cell structure and dynamics. Let's take a closer look at the lipid compositions of the two membrane layers. As you can see, they are different, which is quite important for their function. For example, in red blood cells, or RBCs, phosphatidylcholine and sphingomyelin are predominantly found in the outer monolayer. These lipids help maintain the cell's structure and interact with the environment. On the other hand, the inner layer contains phosphatidylserine, ethanolamine, and various glycolipids and proteins. This asymmetry is not just structural. It plays crucial roles in cell signaling and membrane dynamics. A particularly interesting aspect is what happens during apoptosis, or programmed cell death. Phosphatidylserine, which is normally on the inner layer, translocates to the outer layer. This flip acts as a signal for macrophages to recognize and engulf the dying cell, facilitating cleanup and preventing inflammation. This asymmetry of the lipid bilayer is fundamental for cell function and communication. It highlights how the membrane isn't just a barrier, but actively participates in signaling and cellular processes vital for health and disease. Now let's take a closer look at glycolipids, which are a key component of membrane lipids. The core structure here is the sphingosine backbone, which provides the foundational framework. Attached to this backbone, you'll find a fatty acid chain, adding to the lipid's hydrophobic character. On top of that, there's usually one or two sugar moieties attached, which are crucial because they determine the specific type of glycolipid. For example, cerebrosides and gangliosides are common types that differ mainly in their sugar content and complexity. These glycolipids are essential for cell membrane structure and function, especially in neural tissues where they play roles in cell recognition and signaling. Overall, glycolipids, like cerebrosides and gangliosides, are vital membrane lipids that contribute to the dynamic and complex nature of cell membranes. Let's talk about amphipathic molecules. These are unique because they have both hydrophobic and hydrophilic parts, which is crucial for their role in cell membranes. This dual nature helps improve the stability and fluidity of the membrane, making it flexible yet resilient. Now, when we look at membrane lipids, cholesterol stands out as a key player. Cholesterol inserts itself among the phospholipids and helps regulate membrane fluidity, ensuring the membrane remains fluid enough for function, but stable enough to protect the cell. This balance is essential for maintaining proper cell function and integrity. So, in summary, incorporating amphipathic molecules like cholesterol is fundamental for keeping our cell membranes dynamic and stable, supporting the overall theme of membrane structure and function. Let's take a closer look at the peripheral membrane proteins. These are the proteins that are attached to the membrane but not embedded within the lipid bilayer itself. They play crucial roles in various cellular functions. For example, they include hormone receptors, which are essential for cell signaling and communication. Enzymes are another key type, facilitating chemical reactions right at the membrane surface. Transporters and channel proteins are vital for moving substances across the membrane, maintaining the cell's internal environment. Recognition and adhesion molecules, such as glycoproteins, help cells identify each other and stick together, which is important in tissue formation and immune responses. All of these peripheral proteins work together to support the cell's overall function, highlighting how dynamic and interactive the membrane is. They essentially act as the cell's communication and regulation hubs, connecting the cell to its environment and other cells. Glycoproteins play a crucial role in blood grouping, as they are the main components of the antigens found on the surface of red blood cells. These glycoproteins determine an individual's blood type by presenting specific molecular structures that are recognized by the immune system. For example, the ABO blood group system is based on the presence or absence of particular glycoprotein antigens. Understanding these glycoproteins is essential not only for safe blood transfusions, but also for understanding how the immune system distinguishes self from non-self. By studying these surface molecules, we gain insight into immune responses and compatibility, which is a cornerstone of transfusion medicine. So, in essence, glycoproteins are the molecular basis for blood grouping, linking the biochemical properties of blood cells to clinical practice. So, why is cellular compartmentalization so important? Well, it essentially allows different parts of the cell to perform specialized functions efficiently without interfering with each other. This organization is key to maintaining the cell's overall health and functionality. Now, moving on to how membranes form. It's fascinating because it all comes down to the amphipathic nature of membrane lipids. These lipids have both hydrophilic, or water-loving, heads and hydrophobic, or water-fearing, tails. This dual nature causes them to spontaneously arrange into a bilayer, with the hydrophobic tails tucked away from water and the hydrophilic heads facing outward. This self-assembly creates a stable, flexible barrier that forms the foundation of cellular membranes. So, understanding this amphipathic characteristic really helps us grasp how membranes not only form, but also function as dynamic, selective barriers. If you have any questions about this process or its significance, feel free to ask.